Here, Susanne is pouring the unknown solutions we needed to test the pH values. Next, Emily is carrying out the 2-point calibration for the pH meter. Off camera, she removed the electrode from the storage unit and rinsed it with distilled water. Then on camera, she began calibrating with a solution of pH of 7 by placing the electrode in the solution. Next, Emily takes the electrode out and rinses it with distilled water before finishing the calibration by putting the electrode in a solution of pH 4. In this segment, Lyana is seen testing the pH of her unknown solutions, the first of which was 3.58. She is then seen performing the proper procedure and test the pH of another solution, which was 11.01. In the following segment, Leah is seen testing the pHs of the other solutions. She is seen following the proper procedure and continues to test the solutions. Susanne records each pH reading as Leah completes each test. You can see each of the solutions we tested and their following pHs. To answer the buffer action question, a buffer is a solution that can resist pH change upon the addition of an acidic or basic component. It is able to neutralize small amounts of added acid or base, thus maintaining the pH of the solution relatively stable. This is important for processes and/or reactions which require specific and stable pH ranges. To explain the difference between a weak and strong acid, a weak acid is an acid that doesn't dissociate into ions very easily and can be reversed. However, a strong acid dissociates completely. In order to quantify the relative strengths of weak and strong acids, we can look at the acid dissociation constant Ka, the equilibrium constant for the acid dissociation reaction. The equilibrium expression is a ratio of products to reactants, so the pH is related to the H+. The pH of the solution will be a function of Ka as well as the concentration of the acid. The pH decreases as the concentration of the acid and/or Ka increases. This concept is seen in these graphs. When the strong acid, the acid that will dissociate fully, is paired with a weak base, the equivalence point is lower on the graph. However, when the weak acid is paired with a strong base, the equivalence point is higher because there is more base that is acceptable to accept protons from the hydrogen ions. In the last segment, our group members can be seen cleaning the equipment and lab bench after we completed the lab. Everything was properly stored and put back in the correct storage space.